My name is Daniel Nathans. Welcome to my channel of Science Talks. I'm a famous American microbiologist who won the 1978 Nobel Prize for the discovery of restriction enzymes. Today I'll be taking questions from the viewers, so let's begin. Our first question says, "Hi Daniel, tell us about your background." I was born on October 30th, 1928 in Wilmington, Delaware. My parents were Samuel and Sarah Nathans, who were Russian Jewish immigrants. I was the youngest of 8 children and was born during the Great Depression. During this time, I don't remember much, but I do remember the good humor and warmth that my family had. I began my education in the public schools of Wilmington, and I had my heart set on becoming a physician, not only because I liked natural science, but because my father had wanted one of his children to become a physician. The next question says, "Tell us about your family life." While staying in Bethesda, Maryland, I married my wife, Joanne Gomberg. We had 3 sons, Eli, Jeremy, and Benjamin. I had said that my wife and sons had created an atmosphere of joy and harmony, which was very essential to my career because the medical research field can be very stressful and demanding, and it was nice to come home to family. The next question says, "Describe your research that occurred during your career and education." I had received my MD in 1954 at the University of Delaware, which I had followed in the footsteps of my brothers and sisters. I then went on to an internship at Columbia Presbyterian University Hospital, where I was able to complete my first year of residency. I took a break from that residency and became a clinical associate at the National Institutes of Health, where I was caring for patients in a clinical study of chemotherapy for solid tumors and leukemia. This was a very dark and depressing stage as well for my career because it was very hard to see when the chemotherapy did not work for the patients. This also led me to discover that a protein found in multiple myeloma was being produced from the tumor cells, not other tissues. And I found this out through an experiment where I used mice and added radioactive lysine. This also led me to become fascinated by protein biosynthesis. I also decided to pursue medical research instead of medical practice. I then went on to become a research associate at the Rockefeller Institute, where I worked in Fritz Lipman's lab. I then shifted to studying the transfer of activated amino acids from transfer RNA to a new protein through protein factor. This then led me to accept a position at Johns Hopkins University in the microbiology department. I then discovered that protein synthesis began with the activation of amino acids and their attachment to transfer RNA molecules, which then goes to the ribosome, where the amino acids were assembled into proteins. I then went on to a 6-month sabbatical to Israel's Weizmann Institute, where I was introduced to SV40, a DNA virus. I got a letter at that time from Hamilton Smith, who had said he had discovered an enzyme called influenza bacteria, where I was used to test this to discover restriction enzymes. I had tested Smith's enzyme and 3 others on SV40, which then led me to discover that Smith's enzyme was the only enzyme that worked how we had wanted and cut SV40 into 11 specific fragments, while the others did not. From there, I was able to map and sequence the enzyme, which then led us to the Nobel Prize. The next question says, "Explain the research that went into your discovery that led you to the Nobel Prize in 1978. Well, I was not the only one who won the Nobel Prize in 1978. I was among 2 others, who was Hamilton Smith and Werner Arber. Werner Arber was the one who introduced the idea of restriction enzymes and how they would affect DNA. Hamilton Smith, how I had said, discovered the Haemophilus influenza bacteria, and I was able to test this, where I was able to find that Smith's enzyme did work, and then we were able to publish our research. After this, I also helped formulate guidelines for recombinant technology and became an advisor for the Human Genome Project. I then died on November 16, 1999 from leukemia. Thanks for tuning into Science Talks. I hope you can tune into our next video. Thanks for watching.